Our next speaker today is Dr. Ramon Landin Romero, and he is an NHMRC Senior Research Fellow in the School of Psychology and the Brain and Mind Centre. He's a neuroimaging lead researcher at Forefront, which is an internationally renowned constellation of research groups that use clinical neuroimaging and molecular neuroscience to progress knowledge in non-Alzheimer's dementias and also motor degenerative syndromes. His research develops neuroimaging methods and investigations to uncover the disease mechanisms and neural basis of dementia syndromes. He was recently awarded a competitive NHMRC investigator grant. Big congrats on that. They're very, very hard to get now, to develop the foundations of his research program and create an integrative platform for basic and clinical neuroscientists to work together effectively in their search of a cure for dementia. So I'd like to welcome Ramon. He's going to talk to us about bridging the gap, neuroimaging advances supporting translational medicine in dementia.
Thank you, Leanne. Thanks everyone for having me today. I'm just going to share my screen very quickly. Okay, just going to put the slides on. Excellent. So today I'll be talking about how neuroimaging can really bridge the gap, or try to bridge the gap in between the more molecular aspects and biological aspects of the dementia and the clinical presentations. So this is, of course, an incredibly broad topic, and I'm going to have to cover it very quickly. So I'm really very happy if you want to get in touch, shoot me an email or ask questions in the end if there's time for this. I'll start talking about frontotemporal dementia, which really is a prime example of this incredible biological complexity that is common to really all neurodegenerative diseases. So frontotemporal dementia is associated with the accumulation of different protein pathologies in the molecules and synapses, neurons, secrets, and networks in the brain. Now, this accumulation, progressive accumulation, eventually lead to the development of these major patterns of cortical atrophy or brain atrophy, that in the case of FTD will affect primarily the frontal and temporal lobes of the brain. Now, depending on the actual site of these patterns of atrophy, different clinical presentations will emerge. When the frontal lobes of the brain have been affected, the profile is associated with changes in behavior and personality, and this is called behavior variant FTD. And when the temporal lobes of the brain are affected first, this is conducive of different problems in language, both productions and understanding. Now, frontotemporal dementia are not, and these syndromes are actually not discrete diagnostic categories, but they actually sit on a continuum with different motor and Parkinsonian syndromes that have in common that they are underpinned by similar neuropathological substrates. But the picture is even more complicated than this, because these pathological substrates are actually associated with different subtypes of often co-occurring pathologies. And here, as you can see on the figure in the color, you can see what is the relative contribution of all these different underlying pathologies to the final clinical presentation. And to make things even more interesting, neurodegenerative disease is progressive. That means that even within the same syndrome, we will often see people that will show distinct epicenters of atrophy and incredibly divergent trajectories of brain atrophy over time. So to summarize, the main word here to focus on is heterogeneity. We have this massive heterogeneity of clinical syndromes, which is preventing the accurate diagnosis in vivo of the disease. We have heterogeneity of disease trajectories, which is limiting our capacity to predict and have an appropriate prognosis of the disorders. And we have heterogeneity of pathologies, and therefore this is limiting our capacity to develop effective treatments. So how can neuroimaging contribute to close all these different gaps? By neuroimaging, I'm going to be talking primarily about magnetic resonance imaging, or MRI. It's just because it's more available, it's incredibly flexible, and it's not an invasive method. And since we cannot measure in vivo what is the hallmark feature of dementia, which is the actual accumulation of underlying pathology, what we can do with MRI is measure the brain atrophy that results from the accumulation of this pathology. So we can measure the byproduct of the accumulation of pathology. The main measure here would be the brain atrophy, this progressive loss of structure and function in the cells. And it's important to know that it's actually the spatial localization and the temporal properties of the development of this atrophy. It's what contribute to the final emergence of the syndrome. Now, neuroimaging, it's an incredibly rapidly evolving field. If you blink, there's like next day will be like 20 new methods available for you to map different changes in the structure and morphology and functional changes and connectivity, which give us actually a lot of different tools to obtain biomarkers of this brain atrophy at different levels of the gray matter, the white matter, the vascular changes, and the accumulation of these different biomarkers will in turn lead to the identification of neuroimaging phenotypes. And these neuroimaging phenotypes and the relative importance of all these different neuroimaging phenotypes and biomarkers can be then associated with both the upstream and downstream process in the neurodegenerative disease. So this is an example from a recent paper that we published that is using a combination of clinical examination and longitudinal MRI to provide a better characterization in these syndromes. And here we were focusing in the two variants of PPA that are associated with problems in language production. Now, even though these two variants have distinct epicenters of atrophy at presentation, the actual clinical presentation of the syndrome is very similar, and to the point that even experts in language cannot tell them apart at presentation. So here we try to add new acoustic measures of speech in association with longitudinal imaging to try to improve diagnosis on these syndromes with disease progression. We use this, I don't know what's happening with my slides. There you go. We use this new acoustic measure that is called the pairwise variability index that essentially is measuring the time in between intersyllabic patterns in the pronunciation of different words to assess this over time. And as I said, at presentation, the symptoms were not very different, but we saw a very clear effect that emerged with disease progression, in which patients with the non-fluent variant of PPA showed impairment of the pronunciations of changes in the PVI. And then we associated these changes in the PVI with differential patterns of longitudinal cortical atrophy in these syndromes. And this enabled us to actually identify what are the brain regions that are underpinning the progressive impairment in the pronunciation in these words in these syndromes. And we can associate these different regions with different levels of statistical significance and actually measure what is the magnitude of this effect, which in turn can help us to stratify and select patients in vivo for interventions in trial going into the future. This is just highlighting here, this is a project that was done in collaboration with Professor Kiri Valar, which is a expert speech pathologist. And this also outlines how neuroimaging can really add when combined with different approaches as well. As I said, we cannot actually measure pathology in vivo, but we can predict it using a similar approach, using, in this case, longitudinal imaging to target what are these specific biological substrates. So if we are to develop treatments for dementia, we're going to have to able to map out and to identify what are these different pathologies in vivo. And so we can benchmark interventions as they become available with the patients. And here we use this very valuable cohort that we have with the people that have longitudinal imaging available, but also have pathological confirmation of postmortem. And we divide these cases according to the main pathology present. And as you can see here at the right of the screen, of course, these different pathologies were associated with a very significant clinical heterogeneity. And we mapped what were the cortical patterns of atrophy, both at presentation and with disease progression in the groups, to outline what are the regions that are more associated with the progression of these pathologies before the onset of the clinical presentation. Now, again, we can compare these patterns of atrophy between the syndromes to actually identify what are the cortical sites that are more significantly associated or show the greatest magnitude of effect according to the underlying pathology. And then we can go back and we can obtain these different curves of different trajectories, and we can posit or say like incorporate an individual observation or individual patient in these group level trajectories to anticipate and predict the spread of this atrophy. Another thing that is very interesting is to actually focus, going a bit and step backwards and not focusing so much in the cortical atrophy, but in the structural connectome. So the network degeneration hypothesis is telling us that pathology will spread through these different anatomical connections of white matter in the brain. And we know that the white matter change actually precedes the onset of the gray matter atrophy. Once the atrophy is so massive at the level of gray matter, is often too late. And for this, we're using new methods that are actually developed here in the Brain and Mind Center by Professor Fernando Calamante and his team that used a new approach to map intra-axonal volume changes of white matter axons. So pixel-based analysis, essentially, it's adding a more accurate modeling of different brain fibers in the brain. And this can result in a better, more accurate mapping of the anatomical connectome that can solve different problems such as crossing fibers and the massive complexity of the white matter connectivity in the brain. And it has another advantage, which is that the measures that we can derive of pixel-based morphometry are actually much more readily interpretable from the biological perspective. So we obtain essentially measurements of a cross-sectional bundle of the fibers that tells us different changes in the density of the fibers, but also changes in the cross-section of these fibers, which in turn are biological correlates of processes such as demyelination or atrophy in the white matter connectome. We use these methods to map out the different changes in the white matter connectome in the same groups that I described before. And we obtain widespread white matter reductions in both tau and TDP groups with overlapping frontotemporal regions. But we also were able to determine what are the specific sites of atrophy in the white matter connectome in these groups. And we can take this even one step forward. And instead of moving from tracks, we can actually define different, or we can gain resolution from actually fiber track dissection. So we obtain, using different methods, we can obtain all these different fiber track bundles. And then we can actually isolate these tracks and beam these tracks into different cross-section in the along the anterior to posterior length of the track. That will in turn enable us to actually obtain microstructural changes across all these different beams along the tract. So if we were to focus in the, for example, behavioral variant FTD, and we are isolated here, what are the main changes in the cortex, we can derive what are the white matter tracts underlying these cortical changes, obtain the tract, and then using this fiber tract dissection, we can actually map out where along the anterior to posterior length of the tract, the changes are greater. And you can see here in white, in the comparison between behavioral variant FTD and non-fluent aphasia, how the anterior portion of the tract is being affected significantly in BVFTD compared to an MPA. Now, if we were going to do this longitudinally, we will always be also able to measure how potentially pathology will spread in an anterior to posterior fashion along tracts, and therefore giving us a more accurate mapping of the different disease stages. This is just another study that is done in collaborations with some colleagues at the Brain and Mind Center, and in particular with Dr. Zach Catterton. And here we are combining or trying to combine molecular and macrostructural aspects of neurodegeneration. So as I said before, atrophy is the main feature, one of the main features of neurodegenerative disease, the breakdown of all these neurons and connections. So when this happens, when there's the nucleus of the cells breaking down, there's all this genetic material that goes into the peripheral blood flow, and this is called cell-free DNA. Zach is very interested in mapping these different changes in vasculature and in the spread of these cell-free DNA changes as a result of an insult. And he's developed these different molecular assays that are able to precisely map out the cell of origin of these changes. So he can obtain out of a blood test whether the cell-free DNA is coming from different populations of neurons in either on the white matter or in the glia or in different neurons. Now, this approach is very powerful, and it can be used in combination with neuroimaging to actually provide sensitivity to this method and to validate whether the measures in the blood are actually reflecting what's in the underlying neuroanatomical change. And to do this, we obtain different patterns of longitudinal MRI, and we correlate our changes of atrophy with the changes in cell-free DNA. And what we found is that there is a very nice concordance of results. And as the neuroimaging changes are progressing over time and different regions of degeneration, we are obtaining measurements in the blood and increases of all these different cell-free DNA aspects. And this is a very exciting approach because it can actually lead to a capacity to detect neurodegeneration with a blood test before the emergence of particular syndromes. So just to summarize, accelerating So neuroimaging has a very important role in accelerating the translational medicine in dementia. This is a figure that sort of like puts together all the different things that I've been discussing before. Obviously, it's a massive undertaking, and it will require different things such as access to great and very large, well-phenotyped longitudinal cohorts in dementia, and multimodal neuroimaging approaches, and integrated protocols of neuroimaging techniques that will enable us in turn to obtain or to work out different associations across different levels: the clinical level, the molecular level, and the neuroimaging level. But I will argue that we're actually in a very good position here at the Brain and Mind Center. So I'm lucky enough to be part of this large group of neurodegenerative researchers working in non-Alzheimer's dementia. We do have access to very large cohorts or very well phenotyped and genotype and molecular phenotyped cases of FTD, MND, Parkinson's, and healthy controls as well. We do share similar approaches in the way that we work with neuroimaging and different schools. And we are also now developing an infrastructure so we can automatically ingest and analyze and harmonize all these different data sets that will ultimately help to help us understand what are the different processes in neurodegenerative disease, improve and expedite diagnosis, and help us for treatment development. Very quickly, I just want to thank a lot of people that are actually making this work possible. My funding, of course, my supervisors, my amazing team, the students that have been supporting much of this work. Fernando's in the imaging data service for providing and helping with all this infrastructure development, helping with the development of my job, and also give a shout to all the people in the space, such as Max Shine, Michael Burnett, and Team One. They're doing an amazing job in the neuroimaging space as well. And I will highly encourage you to have a chat and have a look at what they're doing at the moment. Thank you very much. And happy to take questions if there's time for this.
Thank you, Ramon.